Our speaker tonight is Dr. Zachary MacDonald, and his talk is titled Conservation Genomics and Conservation Genomics. Oh, something went wrong there. Okay, I'm sorry. The case of a curiously isolated and endangered butterfly. Zach is an assistant professor in the Department of Entomology at the University of California, Riverside, and his research focuses on insect conservation genomics and biodiversity using whole genome data to understand population structure, evolutionary history, and extinction risk in threatened insects across North America. Most of his work centers on butterflies and damselflies, and he aims to use genomic data to change the way that managers think about the species and ecosystems they work with, translating evolutionary insights into practical conservation decisions. In his free time, he can usually be found driving up and down the western portion of the continent looking for various butterflies, which is how many of us met him for the first time this past summer during our WBA conference. So thank you, Caitlin and David Droppers, for mentioning that there's this guy who's working up in Canada on this curiously, and I was like, oh my God, we got to get him. So here he is, Zach. So thank you so much for being with us tonight, Zach.
Okay, Paulette, thank you so much for that introduction. And yeah, I was just so stoked to meet all of you. That was so awesome. I was in a parking lot just in the middle of nowhere. I don't even really remember where it was, to be
Pearl
honest.
Mountain Ranch.
Oh, okay, yeah,
Pearl
that's where.
Mountain Ranch, just
And,
west of Yakima. Uh-huh.
and I mean, I hadn't really seen anybody in days. All of a sudden, there's one net that just kind of comes out of the woodwork with a person attached to it, and then another one, and then another one, and then 40 of y'all. I'm thinking, well, this must be a thing. So that was really hilarious that we just so happened to find ourselves in the same parking lot. That was really awesome.
And then
And
you joined us that evening
yeah, I
too.
got free pizza.
There you go.
Yeah, that was awesome. And David gave just an awesome talk on the fritillaries. So, cool. Okay. So, yeah, I'm going to talk about just conservation genomics and conservation genomics, kind of putting those two together. It's kind of an emerging discipline. It's been a little bit amorphous for a number of years, and it's kind of, I think, really starting to sh take shape now, but mostly in the vertebrate world and in the plant world. And insects, as we're all really well aware, are drastically underrepresented in most fields, but conservation genomics, I think, especially. So we're kind of doing our best to chip away with that and kind of tackle this one species at a time. And this curiously isolated hair streak is just kind of a little fun story in terms of how we stumbled across this new geographic isolate that likely comprises a species. So um, I guess with that, I'll just launch right into it and share my screen here if I can. Now I'm going to present PDF if it's okay because my PowerPoint's acting a little, it's kind of misbehaving. Um, let's go, You can, okay, let's see if we can get it into. Mhm. Okay, how's this looking? Can everybody see my screen nice and big?
Yep,
Yep.
it's great. Yeah.
Okay, all right. So, this here, just on the left, is our team here. This was mostly done in the lab of Felix Sperling, and so I was associated with him during my PhD. I didn't actually do my PhD in his lab, but just kind of gravitated towards him just because he's the most awesome butterfly guy to ever live. So, and I continue to work with him on a number of projects here. So. What is conservation biology and how do genetics and genomics really relate to that? Well, conservation biology is basically trying to use everything that we know about biodiversity science to save as many species from extinction as we can. And how does something like genetics and genomics really relate? Well, it's an important consideration because we're beginning to understand that genetic problems make small populations more likely to go extinct. And there are some reasons behind that. And I just include all of these code over here. This is what like a genetic sequence looks like. And I mean, if you were to actually look at the genome of the butterfly that we sequence, you'd have to scroll through a screen like this for probably 100,000 years before you got to the end of it. So they're like the data are huge. The lab work is not particularly fun. It's pipetting clear fluids into clear fluids, and you're not really sure if anything's working. So it's not actually that much fun to do. So clearly there must be a reason that we do it. And it's the inferences are just so powerful once we really get to the data. And we're interested in genomics for three main things here. And the first is that breeding among closely related individuals reduces the fitness of offspring. Most of us know that we shouldn't mate with siblings, and but sometimes when populations get really small and isolated, siblings and parents and offspring they mate with each other, and this leads to something that we call inbreeding depression. So that can kind of drive a population towards extinction. And also, when you get successive inbreeding, maybe it's not between siblings, but it could be between cousins and stuff like this, you start to lose your genetic your diversity. And less diversity means that you have less potential to adapt to changing environments, new threats, new pathogens. Basically, for natural selection to work, you need to have variability, variation within your population, and that is all genetics. And also, there's this thing that basically the smaller your population gets, the less effective natural selection becomes because the relative force of just random change, which we call genetic drift, becomes more intense. So that's just kind of as into the weeds in terms of genetic theory that we're going to get. But to kind of put this into a picture here, there's this thing that Gilpin and Michael Soule described, and it's called the extinction vortex. So let's say, for instance, a few 100 or 1,000 years back, we had a healthy population on the landscape here. And then through our interventions, we get habitat loss, fragmentation, degradation, and disturbance. And that makes our population size smaller, right? So that's normally what we think drive populations to extinction. But the genetics are really important to consider too, because once you have that small population, you get genetic drift, inbreeding, the things we talked about, leads to a loss of genetic variation, which means that you have reduced fitness, lower adaptive potential, and increased accumulation of those bad alleles, which means you have lower reproduction, higher death rates, whoops, which means your population gets even smaller. Then you kind of get this feedback loop where the populations get smaller and smaller. So you can almost cross a threshold where once your population gets small enough, you don't actually have to have any kind of an external threat to drive a population into extinction. The genetics will do that on its own. And the example here that I show is uh, whoops Lang's the metal mark here up in the Bay Area. And this is a really small population that's just on the verge. It's about to blink out if it hasn't already. So we're doing similar work on this to what we did on the hair streak, just to like to try to pull this population out of the vortex. Okay, so what's the solution to this abstract problem? It's conservation genomics, which is what I do. And it's, like I said, it's kind of this emerging field that draws on all of these other disciplines and it brings them all together with the end goal of always informing how we manage endangered species on landscapes. So that's kind of the end goal. So if y'all are involved in the sciences or you just really like the bugs, you can probably find the field that you're most interested here. This is why I collaborate with a lot of you and why we all kind of collaborate together because no one guy could do all of these different things. So it takes a lot of big teams to kind of bring this all together, which is another thing I really like about this field. Okay, so on to the actual cool stuff. The half-moon hairstreak is basically the species we sought out to work on. Satyrium semiluna. It's I think in the US oftentimes known as the sagebrush sooty hair streak. It's a relatively drab looking thing, some have said, but I never got that. I mean, anytime you really see one land and you get a close look at it, and like, if you're just close to it or you see it through a macro lens or something like that, they're just fantastically cute things. The banded antennae, the big eyes, they're just fantastic here. And what's most cool about it, not just they look so cool, is that there's this little isolated population that have circled here. Can you see where my mouse is? If you just give me
Yes,
a quick nod.
we
Yeah,
can.
okay. So if you look here, these little squares here are where we tend to find the species. And there's this uh, little population up here that's listed as endangered. Us Canadians, it's quite a rare thing for us, even though it's pretty widespread in the US. So this is in some ways a classic example of a species that just tucks up past like the 49th there. And so we say it's rare and we protect it even though it's doing fine in the States. However, this isolated population is really curious. We had no idea how long has it been here for. It's just this really tiny thing that's isolated on one alluvial fan. Was it introduced by some really enthusiastic collector 100 years ago, or has it been there for hundreds of years or thousands of years even? We had no idea, and it's a really difficult question to ask. Um, and it's really far isolated from any other population that we know about, more than 400 kilometers, which I don't know how many miles that is, maybe about 250 miles. So, and the population is really small. I'll show you a picture of basically the entirety of the population, and it's kind of ecologically divergent. So, throughout the US here, you typically find this species on sagebrush, pretty dry landscapes. The Waterton population here, that's the name of it. It occurs kind of more in herbaceous. Precipitation seems to be higher there, and it's just a little bit more moist, and you don't get the same kind of dry sagebrush landscape. So it kind of tipped us off that this thing's pretty ecologically divergent, which sometimes means, okay, it could have been there for a long time and kind of evolved to live in a slightly different place. So we sometimes see that kind of local adaptation to different environments within species, and it's an interesting thing, and it's important for conservation. So this is a picture of Blackistan fan. It's a alluvial fan. So basically behind me are some peaks, and then as like the glaciers melt and they erode the landscape, all of the material gets deposited onto this really flat fan here, and then the stream kind of works its way across. You can see like sometimes it flows over here, sometimes it flows over here, and that's flat in the landscape. And this is the entire extent of the fan. It's about 30 hectares. And every one of these hair streaks that's in existence at this time would be within the frame of the shot here. So it's just an interesting case. Why is it on this particular alluvial fan and not another one, right? It's associated with lupines, which aren't particularly rare. It may be associated with ants. We weren't really sure, but an interesting question to ask, nonetheless. And so, why did we get involved? Is that this is in a national park and it's a listed species. And so, it began to decline through about the early 2000s. A series of droughts kind of led to a decline, and the population seemed to be just imploding, and it might look like an extinction vortex where the population continued to decline, even though there was no obvious extrinsic factor that really drove the decline. I mean, there was some environmental stuff going on, but it wasn't that intense. And then all of a sudden, in 2017, a fire hits the park and burns half of the fan. And so Parks at this point is freaking out. I think they found about three to five adult individuals the following year in 2018. And we were really unsure, is this population going to bounce back on its own? Or should we consider trying to rescue the species or the population by getting individuals from other populations and introducing them to the fan here? So that's what we call a conservation translocation, where we're deliberately moving individuals around the landscape to basically augment a population in decline and introduce genetic diversity to basically pull it out of the vortex, right? So they're not all just having to mate with their sisters and brothers. So we call this genetic rescue, and the definition of this is just a process that increases a population's fitness by introducing new genetic variation. Now the question becomes, where do you source the variation from? You don't want to introduce something that's genetically identical, or else you're not going to actually introduce new genes to the gene pool. So it has to be from a slightly divergent population, but you don't want it so divergent that they're fundamentally adapted to a different landscape or a different host plant or something like that. So, generally speaking, if we're trying to rescue an isolated population in decline, we look for populations that are a pretty close genetic match. And so we have two populations here. We have the southern BC ones here, and then we have southern Montana ones here. And so Parks approached us and they said, we want you to do the genetics on this to basically see where we should source the individuals from. So we all placed our bets and, we do the genetics and we see where it comes from. And there's a lot of things that we need to basically consider before we start to move things around the landscape. And the first is, what does genetic diversity, just a snapshot of diversity of these populations, look like? When we look at the diversity, we can infer what has the demographic history been. And what I mean by that, that's how large has the population been in the past? There's a lot of theory that says that if a population used to be really big and then it implodes and gets small, it's probably going to go into the vortex and go into extinction, right? Because it has all these kind of bad genes and stuff like that that can be associated with a really large population size, but they don't express the bad genes because the genetic diversity is high. I'm not really going to get too deep into that, but that's just kind of something that we know. If, on the other hand, if your population's been small for a very long time, it can probably continue being small for a while after that because it's probably purged a lot of the bad alleles. So we need to basically reconstruct what the demographic histories look like. And also inbreeding depression, we have to assess, is there even inbreeding going on within this isolated fan here? So, the size of the landscape's quite small, but you could theoretically fit a lot of individuals into that landscape. Typically, you don't get inbreeding depression until you're below like a couple thousand individuals within a population. So it might actually be of sufficient size where you don't actually get inbreeding. So we had to check that out. And now that's a snapshot of what genetic diversity looks like within the populations. Now, what does differentiation look like between the populations? And that's when we start to talk about local adaptation. Can we look at the genes and basically infer, are these things divergently adapted to different environmental conditions? And if they are, if we introduce an individual that's adapted to a totally different area and we introduce it to Blackistan fan, the fitness of that introduced individual is going to be quite low. And if it actually does successfully reproduce, its offspring are probably also not going to be very well adapted. So we call that outbreeding depression. And that's something that we really want to avoid when we're moving things across the landscape. A little bit of divergence is good because you introduce some new alleles that selection can act upon. It might feed on something slightly different, higher adaptive potential. That's good, but too much of it is a really bad thing because it just can't survive in the new environment. So we kind of need to ask this question too, is that are you a Florida panther or are you a Channel Island fox? And I hate to talk about things with backbones, but we just don't have enough examples from the insect world to really make this point. So this is a good example. So Puma concolor corii is the Florida panther, and it is a cat, a Florida cat, and it's been declining for about the last 100 years, and the population was getting really small, and inbreeding depression was like really observable. So there was male testicular issues. They had kinked tails. They had all these kinds of issues going on. The fitness of the kits just wasn't very high, and so the managers of the cats they had to make a decision. Are we going to bring in other mountain lions that are a different subspecies to Florida and introduce them there in hopes that they interbreed and spread their genes? You're going to compromise the uniqueness of the subspecies. It's no longer going to be a pure Florida panther, but at least you're going to have a big cat in the landscape. And they chose to go for that, actually. They introduced, I think it was about a dozen females or so from Texas. And that one-time injection of genetic diversity was enough to bring the Florida panther out of the vortex. It's a really cool story, right? So now, the fitness of the cats is quite high, and the population's beginning to grow for the first time in about 50 years. However, the Channel Island fox, which lived just off the coast of where I am here in California, they're a different story. They also have very small population sizes, but they've been small for a very long time. They've been slowly inbreeding with each other for thousands of years. And when you inbreed, you slowly expose your bad alleles to selection, and you can actually purify the genome or the gene pool such that inbreeding per se is not really that big of a deal. Now, you might look at the genetic diversity of these foxes and say the genetic diversity is so low, we need to rescue this fox now. If you were to bring a silver fox from the mainland and introduce it to the islands, that could spell disaster because you bring in all kinds of maladapted alleles. You bring in also a high genetic load. Really genetically diverse populations carry a lot of bad alleles that they don't express because they're genetically diverse. So you need to know which of the two you are, and we didn't know which one the hair streak was. Should we be mixing it with other populations or not? And the first step in that is you need to build a genome assembly or a reference genome, we sometimes call it. And basically, a genome is all of the DNA within a species so that's basically the blueprint on how to build the species. And they're really long. It's like a long genetic map that shows the identity and location of every single base pair. And there's 1.25 billion of these things. So the genome was actually big. And at the time that we sequenced it, it was the first for the species, the genus, and the theclinae. So these resources are starting to be they're starting to be produced more, but they're still a relatively rare thing to do. So we did I think they did the Human Genome Project in like early 2000s, and it was in the trillions. And now we did this species for only about 20 grand. And in the years since we've done this, I think we can get a genome assembly down to about 5 grand. So it's getting cheaper and cheaper all the time. And so you're seeing more and more of these resources pop up. The figures here, I'm not really going to walk through it. They're just really cool, and I made them, so I wanted to show everybody how colorful they were. But the important thing to know is that we have a genetic map for the species. That's the resource that we need to start doing conservation genomics. Now, what do we do with that? So here's what the genomic map looks like. So here is a chromosome, and then here are all of the individual base pairs. So now we have the resource. We can go and collect individuals from across the landscape. Now, the point of our initial collection of these 15 individuals here was just to take the genome out for a test drive to see how well we could align reads. So what we do is we collected 15 individuals. We break the genome up into really small chunks, and we sequence the crap out of them, millions and millions of reads. And on average, we can cover the genome to a depth of about 25 times. So they really stack up. We sequence them a lot, but they're all mixed up and we don't know how to map them all. So that's where the mapping step comes in, where we snap all the reads together so we know what reads correspond to which area of the genomic map that we made. And then we can see where they are different. And when they are, we call that a single nucleotide polymorphism, or a SNP. And so this is basically the areas of the genome where individuals show some kind of variation between the populations or even within the populations. And this is after we filtered. And so before that, I think we were up around 10 million SNPs. So it's just amazing with the sequencing that we do now. In the genetics days, you would be maybe looking at 3 to 10 variable sites that maybe would help you tell between species, but the power of the data, it wasn't that high. When we move from genetics to genomics, we're considering the whole genome at once and all of the information it packs, and it really is vast. I mean, so even though we have very few individuals, we have a lot of information per individual, which means that the analysis we can do are quite powerful. So what do we do with this? There's a statistic called FST, which basically says how different are populations. It compares the amount of genetic diversity between populations versus within. And if you have a lot more variation between compared to within, then that means that the populations are very divergent. And so these FSTs here, they range from about 0.29, round up to zero point 0.3 to 0.425. This was cool because we kind of placed bets, and the BC population here in geographical space is slightly closer to the Alberta one. But of course, you have to go up over the continental divide, which is really high. And so there probably hasn't been a lot of gene flow or exchange of individuals between these populations in a long time. The Montana ones down here, even though they're a little bit more far away, it's possible that as the Laurentide ice sheet kind of moved up and down, that maybe there was some exchange of genes here. So that might be why the figure here is a little bit smaller. But the important thing to pay attention to is just how big the FSTs actually are. There are some groups out there that say if you're above an FST value of 0.25, you're probably a different species. I don't think any card-carrying evolutionary biologist would ever use a firm threshold. It just doesn't exist. It's not real. You have to understand FSTs within the context of the species and what you observe on the landscape. But regardless, the FST values are really big. And this is the first time where we looked at this and are like, oh, holy shit, we might actually be observing different species here, not just different populations. So there's this thing called principal component analysis, PCA, which is just basically where we plot the genetic diversities of the populations out on the landscape, and we see, do they really mix up together or do they really cleanly sort out? And here they really cleanly sort out. So the first axis and the second axis here, they explain a lot of the variation that we observe, whereas something like this that you see some spread, the variation that explains is actually really low. So we don't really, this is the important one here. And we see that they really cluster out. If it's a within species analysis, typically we see the points a little bit more on top of each other. So this is the first clue. Like at this point, I was kind of thinking, okay, these might be subspecies, but we're not really sure. Maybe we can still mix them because, like, the Florida panther, that was a subspecies thing, and they mixed them and it worked good. So we were still kind of thinking that we could maybe rescue the population by moving individuals from another one to it at this point. And then we have another thing that we run, and it's called structure. And basically, it looks where the major population splits are in the genomics. And so the first big split is, of course, east and west of the continental divide here, where we see the populations or the individuals that are on the east. They form one group, and the populations on the west, they form another one. And then when we run things to the east of the divide, of course, Alberta and Montana, they split out very clean. Now, what this is here is that if there's gene flow between populations, then that means if they interbreed with each other, even very infrequently, once a decade or something like that, once a century even, you get slight mixing of these bars where it's not all just one shade or the other, right? That's what we call admixture. We don't observe any substantial admixture. This means that basically no gene flow is going on here to the Alberta population. And so most of what I've talked about, we could just get done if we were looking at traditional genetics using AFLPs or microsatellites or AdSeq or something like this, if anybody's really into genetics. Some of the older style techniques, we might have been able to get this far. The inferences, they wouldn't have been this clean, but we would say, okay, these things are very divergent and we have to proceed carefully. Now, what happens if we look at genetic diversity? So there's a statistic called heterozygosity, and basically that's a measure of how much diversity you have in the genome of a single individual, right? And so we look at the genome, we line it all up, and you can say, okay, we have diversity here, 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 and here. And then we take small averages, right? And then we see basically how much variation we observe in the genome within the windows here. So you can see if we were to look on the chromosome here, maybe we have a het here, 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 and here. So we have a lot of them. So heterozygosity within the window here is relatively high. We move over to the next one. Maybe we only have one het. It's quite low there. Then we take an average across all of the windows and we just get a statistic. So 0.083 is the heterozygosity for this particular individual from the really isolated Alberta population. And we do this for all 15 individuals, right? So this is what one individual looks like. This is what a BC individual looks like, and a Montana one looks like. Now, what happens when we compare the averages here? We see that the Alberta population, the genetic diversity is really not the variability between the individuals is not very large, right? Um, and also the genetic diversity is quite low compared to the other populations here. Now, if we were doing traditional genetics, this is where we would leave off. You would say, oh my God, Alberta genetic diversity is half of what we see in the other populations. The house is on fire. We need to move these, individuals now, or this thing's going to go extinct because most of us know that low genetic diversity is a bad thing. You just need high genetic diversity for a population to be uh, healthy. And now with the genomics, though, we can look at more statistics that really tell us a little bit more what's going on here. And one cool thing that we do is this thing called analysis of runs of homozygosity. And these are sections of the genome that are completely devoid of diversity. And the more inbreeding you have, the more runs of homozygosity you have. And we can also look at how long the runs are to infer when the inbreeding actually took place. If you have a high abundance of relatively short runs, that means that you have a very long history of inbreeding. You've just been inbreeding for a really long time. If you have a really high abundance of really long reads, that means that inbreeding has really recently ramped up, but it wasn't historically as big. So it's just kind of cool that we can look at these kind of statistics and infer what's been going on through deep time. And look at the disparity here. So this is the Alberta population. These are the BC populations here, and this is the Montana one. When we look at this, depending on how we filter the SNPs here, almost a third to a half of the genome is contained within these runs of homozygosity. This is probably the most inbred natural population anybody's ever looked at. It's just amazing. They've been mating with each other, their sisters, stuff like that, for presumably a very long time. So that was a cool, inference that this isn't necessarily a new thing. And so, okay, the plot kind of thickens a little bit here. They're inbreeding a lot, they're isolated, but this is kind of a hint in the direction. Okay, they might have been doing this for a while, which, maybe there's kind of calls for some kind of revised theory here. And this is the coolest analysis that I think we can do. Using the genome of one individual at a time, we can do some kind of math magic that, to be honest, I don't completely understand. I kind of learned it all and then it falls into my brain the next week. But it's called the pairwise sequentially Markovian coalescent analysis. PSMC is what we just all call it because we can never remember what it stands for. And basically, what this is that we build what a population curve looks like going back through time. So we can't actually look in the really recent past, just the way the method works. We can only start looking back about 5,000 years ago, and we can look all the way back to 1 million years ago. Sorry, that's supposed to be 1 million. I messed that up there. And so basically, it was so cool to do an analysis like this because it runs on a computer that's about the size of this room for weeks, and then all of a sudden you get these curves back and you look at them, and you're the first person to know that, oh my God, let's look at the Alberta population here. It's been really small and flatlined for 40,000 years, right? And so all of the curves here for all the populations, sorry, I'm just going to move this little box. You probably can't see it, but it's my Zoom box. I got to get it out of the way so I can see what I'm looking at. So let's start at about 1 million years here. Presumably, it's all one population. The species is emerging here. Then all of a sudden, something happens around 40,000 years ago, and this isolated population splits off, and it's only about 1,000 individuals. That's so cool to be able to basically rebuild what a population's histories look like. And a lot of these curves, they really shoot up here. That means at the end of the Ice Age, a lot of the isolated populations here became really well connected. Sorry if I didn't explain it. This is effective population size on the y-axis here. So basically how big the population is. So this shows us here that connectivity really increased. These populations were all mixing with each other. This one was not. It's been small for a long time. So even though it's small today and super isolated, it's nothing new for these guys. They've been doing it for a while. And it's really cool when you can just look at what's been going on in geologic time and infer something. you plot the last glaciation here, and it coincides perfectly with these curves here. So it's probably a contiguous metapopulation network, and then the Laurentide ice sheet 40,000 years back, scraped across the continent, and not exactly where the Waterton population occurs, but in that area, we know that there was a lot of isolated refugia, right? And these are these tiny little rock areas, oftentimes on the top of like, a mountaintop or something, where the glaciers moved around, and a lot of smaller things like insects and plants, they persisted in isolation. Probably the vast majority of species that were kind of confined to these, they went extinct because of the extinction vortex and things like that. But a very small minority, like we're probably talking tenths of a percent, survive. And this hair streak was one of those that probably survived and then probably bounced around in the area and today is on this Blackistan fan. So it's been there for a while. Those are the genetics. Now, what does it look like when we look at ecological associations? We run a lot of these things called species distribution models. We call them SDMs, and people also call them habitat suitability or niche analysis here. And basically, the point of this is that we look at all of the occurrences that we can. And this is where the community science really comes in huge because anytime anybody puts an observation on like iNaturalist or something like that, I pull all of those and they're so useful for training the models here. The data are truly invaluable. And we look at where are we seeing the species and what do the environmental conditions look like there? And we can build an equation to basically predict across the landscape where is the species most likely to occur? So green is good, white is bad, red is in the middle. We can see that we have a prediction here. So this is kind of in the northern extent of the range. I can find the analysis here just to consider the populations that are relatively close to the one that we're interested in. The model works really well. So we have all these kind of math statistics that we do to show that it does a really good job of splitting where the species occur from where it doesn't. So the model performs really well. It really predicts where the species occurs well, except for Alberta. It predicts that there's about a 0.1% chance that we should find the species there based on the ecological associations of the rest of the species. So this is when we're like, okay, we're really dealing with a fundamentally different, animal here. There's this thing is like adapted to a totally different combination of environmental traits, and summer precipitation is the big one. It's about twice as wet there as it is at any other occurrence across the landscape. And so this is when we started to think, okay, these are deep level divergences. And meanwhile, this is taking me kind of 2 years because I'm a postdoc and I'm dragging my heels, and Felix is like, where are the results? All this stuff. What's going on with the population in the background, right? It was really declining. Did it blink out of extinction? No, it actually recovered on its own. And so this was really cool. And it's a telltale sign. If basically a population used to be very large and you neck it down and you hold it down for a while, inbreeding depression and the vortex kick in and they can't bounce back up. These ones did bounce back up on their own. So that shows on its own that even though there's inbreeding, you're not getting inbreeding depression because this thing's just really used to being small and isolated on its own for a long time. It's been just kind of like hovering on the brink of extinction for a while and has developed a gene pool that's very robust to that kind of thing. So what do we do with the problem of genetic rescue? Just to recap on the things that we talked about, from a genetic diversity perspective, this population is like super low, like lowest you could possibly imagine. Genetic diversity is low. 10 years ago, based on all that we knew about conservation, genetics and stuff, we would be freaking out. We say, we need to rescue this thing right now. We've doomed it to extinction for something because we caused a fire or something. But the effective population size, we know that the genetic diversity has been small and it's been isolated for 40,000 years. And inbreeding depression, we observe inbreeding. We can tell that based on the genetic distributions, that we looked at. But is there inbreeding depression? Is there a negative consequence to the inbreeding? We haven't observed any. It's very difficult to observe inbreeding depression in an insect because it's not like a mountain lion where it's an easy thing to tell. Okay, this one had that many kits, and this one had that many kits, and this one has a kink tail, and that one has like a testicular issue. We can't do that for these bugs in the fan, but we can tell that this thing imploded and then it exploded again. And if you were suffering from inbreeding depression, you wouldn't be able to do that. Now, would we risk outbreeding depression if we were to mix these populations? Almost certainly. If we were to take individuals from either BC or Montana here and we were to plop them on the fan, they would be like, what the heck? I'm not from here. I'm not going to mate with these guys. Or if they did, their offspring would be really maladapted. They don't even feed on the same host plant. It's a different lupine species. We've learned since then that they associate with different kinds of ants. And so basically the maladaptation would be significant. So we recommended let's not mix these things for now. It looks like this thing's been doing okay, and it might continue to do okay in the future. However, the adaptive potential of the population is quite low. So if the environmental conditions continue to change at the fan, precipitation is like all over the place these days. The summers are really hot, fire's an issue. So how do we really conserve this, population if we can't mix it? We can talk about that in the discussion after because, we're not really sure, to be honest. Um, but then this brought a follow-up piece here. All of the work that I've described was in a paper published in Molecular Ecology, and then we had a follow-up note on Zookeys, where we evaluated does like all that we know from asking these conservation genomic questions, should we just give this thing a new name? And as some of us might be aware, naming species or even deciding what a species is a really difficult thing. You have all these different species concepts. Some say that you can only call something a species if you can demonstrate that they can actually breed with each other and produce viable offspring. That's what we call the biological species concept. It's an old one. These things are never going to come into contact with each other on their own, at least not for thousands of years. And we're never going to mix them because parks will never let us. So that's not a testable thing. Morphological species concept, they are kind of morphological, differentiated, phylogenetic. There's all these different metrics that we can basically what we call species concepts that each have their own definition of a species. There's a new way of thinking that's called the general lineage concept of species that says there aren't different species concepts. We only have a single concept of species, and we have multiple different species criteria that we can use to diagnose those. They don't all have to be checked off because you can't always test all of them, or sometimes like all of them are just not applicable. But if you get most of them, you're probably different species. So like let's imagine here you're one species, you evolve through time, and then you start to get phenotypically distinct. So that just means like morphologically divergent. You don't mix genes, you can basically tell each other apart and to only mate with your own kind and stuff like that. You're ecologically distinct. You live in a different climatic regime. We basically went through the checklist for the hair streak here and said, how many of these can we get? Is it reproductively isolated? I mean, we didn't test it, but it probably is. And more from a functional perspective, in that you might be able to reproduce these things in the lab, but if you reproduce them, those offspring are going to be so maladapted to some new environment that they're not really going to do very well. And they're so separated in geographical space. That one's a tough one. Are they ecologically distinct? Entirely. We saw from, the SDM that we predicted there's a 0% chance that the half-moon hairstreak should occur on the Blackistan fan. And then, yeah, so we kind of go down the list. We check most of the boxes. And I kind of wanted to describe it as a subspecies because I didn't want to put a nail in the coffin of this population in case we were wrong. I knew that if we describe it as a species, Parks Canada is going to be so proud of their butterfly, and they're never going to let anybody mix it ever because you're going to jeopardize the purity of the lineage. They're all about this ecological integrity bullshit, as if we can defer to some baseline in the deep past and say that was the right species composition, that was the right gene pool, and we have to do everything we can to defer back to that. We live in a new messy world where things are getting mixed up all over the place, and I'm like, there's kind of a way of thinking that we do have to try bold solutions like hybridizing things, just like we did with like the panther to get it to persist. And so I was really worried that if we described it as a species that parks would never approve us doing any kind of work on it. And not from a fact that, we want to do more work. Of course we do, but just that this could actually jeopardize the conservation of the species. Reviewers really push back on that. You shouldn't think about conservation implications when you're describing species or not. You should just do pure science. Is it a species or is it not? And let the cards fall out and how the social sciences of conservation deal with that, deal with the information. So we were pushed in the species direction, and ultimately it was the right call. And I was kind of surprised that it was well received by the community. I thought we'd get kind of lit up for this one, splitting stuff off. But most of everybody seems to more or less agree with us, which is cool. And a lot of news outlets, picked this one up. So it was the busiest week of our life. Everybody thought it was really excited. This is the first butterfly species, I think, that is endemic to Canada. There's subspecies that you find up there, but I can't think of another species that's found there, and no one else and nowhere else on Earth. And everybody just really seemed to like the story that we could rebuild what the demographic history looked like, that we knew that this thing was isolated for 40,000 years. So it's kind of a very wandering, meandering tale in terms of how we eventually ended up discovering a species when we just set out to mix some populations. So I guess with that, I hope we're on time, but I'd like to thank everybody for sitting through it, and I'd be really happy to just have a big conversation in terms of what everybody thinks.
Can I go?
You
Yeah,
go.
All right.
God.
So you covered all the bases, bro. I had a bunch of questions coming in. I had some that rose, and you answered all of those. I want a copy of that PDF about the species concept and that lineage thing. That's just beautiful. But
Yeah, of course. Yeah.
I'm excited. I was excited when it was described, and now I know why. So I do have one question, and I'll leave it and maybe come back if I have another one on the way. So you are familiar with the Laurentidian and Cordilleran ice sheet interface, which is said, at various times by various people to form a corridor between the Arctic and, or at least to some degree, a corridor of
Yeah.
unglaciated
That was
area.
a split,
It
yeah.
looks to me like that's right in the middle of it.
It is, yeah. The corridor was probably a little bit to the east there. So when it split open, that was like 14,000 years ago when it first split or something about that. And it was a little bit east of the divide, I think. Like it kind of ran through Edmonton and then between Calgary and the mountains, I think. But I'm a little bit shaky on that, so correct me if I'm wrong. But yeah, that's right in the mix of it, and that's probably where this refugium was. But I really need to brush up on my glacial history. So if you have any kind of insights, I'm very much,
Oh, I'm full of stuff, but it's not necessarily insight.
yeah. Awesome.
May I ask you a question, Paulette?
Everybody can unmute and ask questions. So yes, please, Bob. Yes, thank you.
Hello, Zach, and congratulations on beautiful work,
Oh,
wonderful bit of natural history
Oh,
and
awesome.
science, and
Thanks.
also a very, very cool name in the lexicon.
Yeah.
Very, very cool. I love it. Couple of questions. One of them on your circle chart you had of the many different disciplines involved.
Yes.
One that was missing, and I am one, so I noticed it, was biogeography. I think
Oh,
you might
yeah.
reconsider casting it with biogeography and faunistics as one of the major disciplines here. You could include it under natural history or others, but really it is at the bottom because until we properly discern these things in the field, where they are, where they're not, obviously this work doesn't happen. And that, of course, becomes much more complicated and possible with iNaturalist and other modern tools. But I studied what I studied in my doctoral work was ecogeography, the blending of ecological technique and application and applied biogeography. And
Mhm.
I think that's a very, very big part of this because this is simply one of the most spectacularly relict species in the country, if not the world.
Yeah. No, I think you hit the nail on the head, and it's so close to home that I didn't even see it because it's one of those things that's so central that it doesn't even come to mind because
That you're
to me, it's
exactly right.
almost synonymous with biogeography. So the field, we kind of call it landscape genetics
Well,
sometimes.
I wondered if you weren't kind of including it under that, yes. But
Yeah,
I
so I
wondered
should
if,
make, yeah, yeah. No, in the next group I give this talk to, there will be a biogeography.
perfect, thank you. And while we're on the subject of endemism and relictualism, uh, there was one widely accepted, fully endemic Canadian endemic for many decades, and that was Papilio brevicata in the
Oh,
Maritimes. However, it has been discovered in Maine in 2 or 3 sites now. So
right.
now
Yeah.
it's US as well. Too
Yeah,
bad,
it's
too
speaking.
bad.
Yeah.
That was a very, very cool Canadian endemic.
Yeah, I've never seen one, but
Me neither. I was going last summer and trip got called off. But I still want to see that butterfly. I
no
didn't get to
kidding.
see it on the butterfly big year.
Yeah.
The last thing pardon me. But those people in Maine, there are some very good lepidopterists,
I'll
Maine
bet.
and deer in those, and they're working with Maine and the Maritimes as if they are one bioregion, which they are. And
Okay.
so it makes sense that brevicata would have popped up in northernmost Maine.
Uh-huh. Uh-huh. Yeah.
The other thing I had is this, with respect to possible habitat measures, absolutely agree with all your conclusions so far, and and I'm really very happy to hear about outbreeding depression. I knew about the concept, of course, but I'd never heard the term actually stated, and it's so important because everybody knows about inbreeding depression, but
Yeah.
very few have thought or written about the dangers of introducing inappropriate genes. Uh, so you took care of that.
Yeah.
But 2 questions. So the discovery is in a national park.
Yeah.
In that sense, it resembles the discovery of the island marble in Washington State,
Right.
uh, in the San Juan Island National Historic Park, and that's a great boon because you don't have to try to buy it from oligarchs or anything. But of course, it introduces its own whole management formulae.
Hmm.
And so I'm wondering, is there any particular management regime on the fan, or is it strictly laissez-faire at the moment?
Yeah.
Apparently, you gotta burn them. Apparently, you gotta burn them.
Yeah, you got to burn them every so
Well,
often.
maybe
Yeah.
so. The
Um,
question I have, and I'm going to have to run because I locked my key in the car and locksmith's coming, but I want to ask you, is there any other potential habitat that has been discerned or possible introduction to make a second population?
yeah, you hit the nail on the head. So there are management on the fan. Spotted knapweed is the big issue there. So all of
Right,
the parks control goes towards controlling that.
right.
We're kind of the camp that like spotted knapweed is the worst thing to ever happen to the species, but they've not done. I'm really trying to push them. You have to do the analysis to show it that they're competitively excluding the host plant because it seems to me like they live in slightly different parts of the fan.
Yeah, plus it's a damn good nectar plant for a lot of butterflies.
Right. Okay. I didn't actually. Yeah. So I
It
haven't
is.
seen the hair streak on the knapweed itself, but
Yeah.
yeah, we are actively the only good conservation strategy I can think about is to try to establish a secondary auxiliary population.
Yeah.
Parks Canada have this ecological integrity thing that they're really into, that they say that it like a thing has integrity when its parts and processes are characteristic of the natural baseline or the region. And the problem is that if we discover a secondary fan within the national park that never had the hair streak before, and we introduce the hair streak there, even though we're doing everything we can to reduce extinction risk, Parks is going to probably come back and say that we undermined the ecological integrity of this new fan by introducing an unnatural hair streak, so like an invasive species at a very micro scale. So I'm just fighting that concept a lot, and we're trying to go after it like some of this manipulation has got to be okay if we want to keep these things around, right?
Well, they have certainly done a lot of manipulation for other
Yeah.
species in other parks. There's going to be those purists, yes, and they may be a roadblock, but you just got to find the progressive, the thinkers in the Park Service,
Oh yeah. No,
Who
and
about
there are a huge faction. I'd say that the majority of them are really card-carrying biologists who, they want to do these things, but there's white papers and all these kind of, mandates that we have to work around. And so that's why I want to say that the strength of the paper though was actually collaborating with Parks through the whole part. It's not like we did the study, in isolation and then presented the results to them and then tried to convince them. They were a part of every step in the analysis, and that
Right.
was so powerful for they like they were right on it, and they had the signs deployed before the description was already out. They were like they were right on this thing. So the people that we work with in parks are absolutely fantastic. But
That's great. And they must be so grateful to you, really, coming up with this cool animal in their park. They got to work with you a little bit. And
yeah, yeah, we'll see. But yeah, I mean, we all want to see it saved, right? So, but
exactly.
yeah,
Thank
no,
you so much for a
yeah, thanks.
wonderful program and a
Yeah,
wonderful piece of work, both in genomics and in conservation, both.
and biogeography.
That's right.
Thanks, Bob. It's really nice to see you and meet you.
You too.
Okay, feel free to unmute. Any other questions? I've got a couple, but I want to open the floor to anybody else. Please feel free to unmute, or if you don't want to do that, put it in the chat.
Well, one more thing. Have you talked to Nick Grisham?
No, not about this. No, no, I haven't. But
Keep
I
it that way.
know.
Just be glad you got your name done first.
Yeah. Yeah, no, yeah, it's always interesting. Like, trying to keep up. There's a lot of work that comes out of that group. And even on the metal mark example that I gave, there was a paper that just came out a couple weeks ago where where that group has made the decision that the Langai small population is actually a much bigger California clade and stuff. So we have a really different I come at things from the population genomic end, and then sometimes we accidentally describe species or find things. He comes at it from the phylogenetic end, building trees, and defines the monophyletic groups and sets like FST thresholds. They're very different perspectives in terms of how you diagnose species.
No kidding. 0.25, baby. 0.25.
Yeah, okay. Yeah.
Species, genera, tribes, families, 0.25.
Yeah, yeah, no, I was really sad to see them sink Barry the Parnassius in the Sierra here, just based on a threshold without a coalescent-based analysis of these kinds of things. It has big implications for field guides and conservation and stuff. So it's just, but I'd love to chat, with that whole group and see what they're thinking.
You would.
Yeah.
Just
Trust
take,
me, you wouldn't.
just take Felix with you and you'll be okay.
Yeah. Oh, Felix is, yeah. No, I'm definitely in the Sperling camp.
Yeah, I think most of us are. Hey, by the way, it's also got to be probably, I mean, common names, there are different methods for adjudicating them and so on, but it's probably going to be maybe the longest common name in the North American fauna. Yeah. Curiously isolated hairstreak.
I didn't think about that,
Yeah,
but
maybe the longest, but surely the coolest.
yeah. Well, Sperling will be glad to know that because he's always, going after me for my pedantic, like the sentences and all the stuff he asked at it and stuff like that. So if he knew that I named the longest thing in the country, he would find that very funny. So I'll have to ask him.
Well, I took care of that by just naming a subspecies Sperlingi, so he couldn't have a problem with that.
Okay, fantastic. That's great. And so the next step of the work too is that we got to build this out and do landscape genomics of the entire species complex, including Phylogenosa. So I've spent the last couple of years kind of driving around trying to fill in all the gaps. Caitlin, I think, pretty much quadrupled my Pacific Northwest representation. So
I was gonna ask, like, where you still have gaps and in like Washington, Oregon that I might try to fill in this summer.
Yeah. Let me send you, I gotta plot them all out and then I'll send you and we can maybe look at the distributions and stuff like that and see. But one other unintended consequence of describing this thing as a unique species is that the Calgary Zoo and Parks Canada to a
Okay.
certain extent, have maybe lost a little bit of interest in the broader species complex because they're really concerned with the fan. And and the fact that that is its own isolated species to some suggests that you don't need to know what's going on in the rest of the complex. The other thing I like about the general lineage concept is a species description is just a hypothesis and it's out there to be falsified. We could be totally wrong. We could discover intermediate populations with more search effort between Montana and Alberta. That would falsify our ideas or our species description. And so we need to continue work on the whole complex to really get a good feeling, and we can identify all kinds of important adaptive genes and stuff like that within the broader species complex with important implications. So if you ever talk to anybody who's involved in the big conservation decision making, the broader genetic context is really important for us.
Well, semiluna just barely gets into British Columbia.
Yeah. Yeah, it just barely tucks up in there. It's and I actually haven't seen those BC populations before. Like I've been right on the border, like Spokane and stuff like that. But
Do you have samples from the Okanagan, the Washington side of the Okanagan? Like,
I have a couple, but I was having a hard time finding them right at the border. I was a little bit late last year.
okay. Because
Yeah,
I go up to the Sinlahican fairly regularly, and that's only 14 miles south of the border, and I easily get them there. So,
yeah, no, that would be awesome. Yeah, I would love to see some there. That would be so cool. Another cool observation that I'd like to share with everybody is that how I find the half-moon hair streaks in the field and differentiate them from Bois Duval's blue, which really look, just about the same unless you have them in the hand, is the blue you can kind of keep your eye on when they're flying around the buckwheat and stuff. These hair streaks, man, they loop around so quickly, I can't even track them with my eyes, and they're so hard to get close to. They're really a tough thing to catch, I found. But the Waterton population, the isolated one, you can go right up to them. And at most, if you scare them, they just fall off their plant. And I think that this is an evolved flightlessness, just kind of like an island bird, and that if you leave, you die. And there's no dispersers that come in spreading dispersal genes. And so it's a windy place too. You don't want to get bumped from your your nectar plant, your food plant, or and disperse far away because you're not going to survive. And so if you get a chance to go up to the fan, they're just really readily observable. Like you could sit there with your, with the camera lens an inch away from the bug, just shooting the same one all day. They're so cooperative and nice. So it's just an interesting split between what I imagine the BC and even the Okanagan ones are. It'd be really cool. So when you're in the field, please do take note of if you can really get close to them. I'd be curious.
What's your experience, Caitlin?
uh, hit and miss, I guess. Like I'll frequently encounter them up on Nanum Ridge, but it's also frequently very windy up there, so they tend to be just hung on to the plants, and I'll get pretty close and get some good photos, but that might just be because it's so windy that they don't want to let go of the plant, like you were just saying. Sinlahican, it's a little bit more hit and miss. I don't have so many photos there. Um, they do tend to zip around quite a bit.
And they're ecologically very diverse just in Washington. The arid subalpine and the east slope, up to 5,000 feet, they could be in open meadows habitats. And then they're right out in the sagebrush.
Right.
Yeah. So, I mean, but they're not uniform between them. They're in local, little colonies. So,
Yeah.
they're not like sulfurs.
Yeah. Yeah, that'd be interesting to see some of those higher elevation ones. I know I was getting them in kind of the interior volcanic range, like, all the mountains hood all the way up. And those were probably the closest because I think precipitation there's a little bit higher and the lupines start to look like kind of getting out of the sage, what you're thinking. Like those are the higher elevation ones you're talking about, right?
Yeah.
Yeah. Yeah. No, that'd be interesting to take a closer look at those.
So,
So,
Allison, did you have a question?
what?
Oh, well, I just wanted to say how much I enjoyed the talk, but also I work on the Olympic Peninsula. And a lot of times when we will say, oh, what species or subspecies do we have here? And the answer is usually, well, it's sort of this, but it's not really. And it's sort of that, but it's not really. And
Yeah,
so that makes me feel like they're doing what you're describing there.
I think so. And it's funny you mentioned that too, because Ross Winton and Erica Henry and there's a number like Taylor Eye up there, the checker spot. I gather it's basically arbitrarily defined with an elevation threshold.
Yeah,
Over
yeah.
this,
The
it's
Edis is high, then you come down and you get the Taylor Eye, but
yeah,
the Edis isn't really all the way Edis. There's something a little different about it up there. So
yeah.
Yeah,
And
the high elevation Edith in the Olympics aren't even in the same group with Taylor Eye. They're more the intermontane group, the so-called Hutchins Eye group or the BNI group.
yeah, yeah. So yeah, we're really gonna because I've just sequenced 370 Ephidrius from California, and we're just gonna work our way north and figure it out because I think there's like some 30 named subspecies within California alone.
oh wow.
Ah, I hope not.
Yeah. Yeah, and then Icarioides, or sorry, Icarisia is another group in the peninsula with a lot of really curious, little isolates, isn't it, Allison?
Yeah, the little I think the Puget Blues.
Yeah.
Yeah, and then some are up, you know, you'll find them at a certain elevation, but then there's this whole like sea level group there, right
Yeah.
by the Strait of Juan de Fuca.
Yeah. Yeah, I'd really like to spend some time up there just getting a feeling for it.
Okay,
Make sure you
Yeah,
contact
yeah.
us. We'll let you know all the good places to go.
absolutely. So any other questions, comments? This is just free discussion time, so please feel free to unmute, ask your question, make your comment.
Did you say 30 subspecies of Edithii in California?
I think that's what it is. If it's not 30, it's high 20s.
Well, making sure we're talking a whole bunch of subspecies of Edithii.
Yeah. Yeah.
That's good. Thank you.
Yeah, Euphydrius, yeah.
Well, you got to keep in mind that California is huge. It's like going from Boston to Atlanta. It's huge.
Yeah.
So it's not like comparing the same number of taxa in Washington, which Washington's tiny. I'm sorry.
Yeah. Well, and to that point, it's not only big, but it's so biogeographically diverse too, right? I mean, you go 100 miles in any direction and you might as well be on another continent. It's crazy. So, but they've done a lot of stuff like even in the Sierra, just weird, elevational bands that like Orlacus is the lowest, and then there's some other one, and then like Nubagina is the highest one, and there hasn't been any genomics or even genetics done to test these kinds of things. So there was, I think yeah, it's just the polyerlic group. They named a lot of stuff.
They're busy. Yeah.
Yeah.
So I was wondering if anybody has thought about climate change susceptibility for this tiny, tiny little population that only lives in one spot. I mean,
Yeah.
it's obviously susceptible to a single wildfire, although it seems like it popped back really well from that.
Yeah, only half the fan burned, which is important, I think. If they got the whole fan, then it might be a different talk. Um, but I think that's what we'll do. I got hopefully a grad student who's pretty keen on the climate change extrapolation stuff. So we've been doing a lot of that to kind of predict how ranges are going to shift. Because I mean, you can either move, adapt, or die, right? And it's probably not going to move, and it's
Not
probably
on its own. No.
yeah. Sorry.
Well, there's also the idea that, sure, maybe the national park doesn't want you moving it, but is there other public land in the area that may have suitable habitat that would be more amenable?
Yeah, and sorry, I shouldn't paint national parks quite in such a narrow light. They would come around. I'm just saying, like, the individuals that we work with are very pro conservation, but parks as a whole has very strict mandates that are difficult to get around, particularly this idea of ecological integrity. So I should just be a
Yeah.
little bit more careful about that. But the difficult thing is that we'd still have to source individuals from the park. Parks has been very cooperative with providing specimens and seeing the value in sacrificing a few old males for the genomics and stuff. So they're clearly bold and they want to try things and stuff. And I think that if we did find trying to even rear this thing, so I'm not sure if David is on the call here, but we gotta we got to talk to him in terms of I think that the Calgary Zoos just have a hell of a time even trying to get these things to lay eggs on the lupines. So we really got to ramp
Well,
up
they
our
probably lay eggs in the dirt, you know.
yeah.
They diapause as eggs, so best tuck them in the dirt.
Yeah, I think that's what they
Well,
do. Yeah, but then do you have to have the ants present there? They're associated with a
Oh,
very
yeah.
specific formica, and the other ones are camponotus. So yeah, yeah,
well, Dave
Yeah,
Nunley is here. Dave, do you have any
of
comments?
captive, there's been a lot of captive breeding with the Taylor's checker spot
yeah.
where they've augmented populations that way.
Yeah, yeah, no, definitely. I think that I would be very much for them experimenting with trying to get a secondary population going. It's just I think they just need to get the momentum. I mean, they don't have the Calgary Zoo, they don't have a rearing facility right now, but they could learn a lot from the Taylor Eye facility, the San Diego, they got a big rearing group down here and just need more flow of information on this.
So Dave
Yeah.
Nunley is here. Dave, do you have any comments about rearing?
Oh, there he is.
Turn the audio on. What was the question again?
Go ahead, Zach.
Well, my Calgary Zoo counterparts are just having a relatively difficult time getting the curiously isolated hairstreak to, I think, even oviposit. I don't think they're getting quite that far. And the problem is that they have a lot of because they're a zoo and the national parks, they have a lot of restrictions in terms of how long they can keep an individual in a cage, but for an observation period before they re-release. And if it was up to me, you leave a female in there until she's just on the verge of death, and then she lays all of her eggs because that's the last thing to do, and then you release her. But they're only allowed to keep them for 6 hours or something like that. And I'm wondering if you think that might be a part of the issue.
Are you following what he's saying, Dave? Okay. He's asking a question.
Dave?
Uh, David James and I reared the half-moon hairstreak in Washington. One population we reared was down in the low shrub steppe, probably about 2,000, 2,500 feet. The other population was up in the mountains at about 6,000 feet. And we found quite a bit of difference in the two populations. Now,
Oh.
that might have been just habitat, but we know consistent differences in pretty much every stage in color and to some extent in the larvae, whether there were the double white stripes down the dorsum or anyway, a number of different questions there or differences there, and we wondered for quite a while if they were the same species, but they pretty much had to be in line with our knowledge at that time.
Yeah, yeah, yeah. So just a lot of variation even within populate. Yeah, yeah.
Yeah.
Well, if you ever want to come up north and try your hand at the curiously isolated, I'm sure the zoo would love to have you.
Okay.
Did you have any other comments, Dave, or questions?
Do you have any other questions, Dave?
I don't think so. Do you, well, do you think that the differences, the two populations you found, do you think that was mostly a difference in elevation, or is there something more to it than that?
I think so because, for instance, in the ecological differentiation analysis that we did, we took every available record and we made a model on that, then extracted the suitability at the records used to train the model. So basically, you get a distribution of, say, that the species is found in habitat suitability values 0.7 to 1. Then the Alberta thing, the isolated one, was 0.01. it was way off the deep end, right? And so we do build kind of a distribution that includes all kinds of different elevations and whatnot that's occupied by semiluna across its range.
Hmm.
I have one more question, Paulette, if that's,
You go ahead, Bob. Absolutely.
well, if somebody
Well,
else.
you have to ask me, Bob.
What'd you say, John?
Never mind.
No, I had to leave for a while because I locked my key in the car with the lights on.
Oh, that's a fun one.
We got a locksmith here. We got to go in the car ran. I drove around the block, so we're cool. Everything's good. Zach, in my absence, did you discuss androconial patches?
No,
Yeah,
no.
because this is a very interesting species, or the
Yeah.
androconial patch. You know,
Yeah,
the so-called stigma, the
yeah,
stigma. And
yeah,
as you know, one between semiluna and the city hairstreak, one phylogenotum,
yeah,
one generally has the androconial patch, one generally does not. But
yeah.
Andy Warren has found populations in Oregon in which some do and some do not, right? Am I right, John? Is that how it
Yes.
goes? Yeah.
Yep.
So what's the deal with the curiously isolated hairstreak? Does it have them or not?
Yeah, the male's got him.
Hmm.
And yeah, I've tried to use that patch to because I'm also trying to include Fuliginosa in the in our assessment. Like in our next more broad landscape, we'll probably sequence like 100 individuals or so, and I've been trying to find Fuliginosa and use that trait, but I just can't tell these things apart, man. I mean, like Fuliginosa going a little bit more brown maybe or something, but and I've been talking to Eric Runquist a fair amount, who he did a master's on this with Art Shapiro,
Oh,
and
right.
yeah, he ran all over the place catching him. But yeah, I'm not sure how we should look into that more in terms of really when we do our landscape genomics, being very explicit which populations do the males have the stigma.
My personal experience with those two species is they're very different in the way they behave.
Yeah.
Yeah, first time I ever seen Fuliginosum was in Southeast at Caitlin and I went to Davis and we went through Southeastern Oregon or Southwestern Oregon and up on the way up to Mount Ashland we encountered some. It was like, whoa! I mean, they fly different. You
They're
know,
a
hell,
lot darker, and yeah, the wing shape was just a little bit different
yeah,
too.
yeah, yeah.
Yeah. Yeah. And they're easier to catch too, I found. I don't know if you found that.
Well, they were right along the road, so that made them easy.
There you go. So there is a question in the chat from Patricia. Do you want to ask Patricia, or if you don't unmute, then I'll go ahead and ask for you. I know sometimes you have issues there. So I can go ahead and start, and if you want to weigh in, that's great. She was asking about the individuals that you were using, whether you use a leg, and I think I know the answer to that, but you also elaborated how you chose the samples. So could you talk a little bit about that?
Yeah, it's a good question. So we use the whole body. We collect individuals whole and we use the thorax. There's a lot of groups right now that are even pulling a leg and they stoke the leg and they get DNA and then they give the leg back to the museum or something. And it's whole genome, but it's whole genome in a very different way.
Oh,
We are doing what we call deep whole genome resequencing, where we have the genomic map. We want at least 25 reads, at least per location in the genome. Typically, these other groups, when they're doing whole genome, they're going for like 1 or 0.5 on average. It's just like skimming the surface. That's more suitable for broad scale phylogenies and like doing highly conserved regions for gene trees and stuff like this. It's not usable for the kind of analysis we do. We need genomic data that are a little bit more thorough. And so typically for building the genome assembly, we take the individual, we flash freeze them. It goes off to a fancy lab that does techniques that I can't do for DNA extraction. And because they need to get really long reads, like 10,000 base pairs. For the kind of sequencing that we're able to do, which is the short read, we either flash freeze them or we put them in, ethanol or a solvent. And but we are experimenting with using legs and building those curves backwards through time. You can cheat with low sequencing depth, but it affects your curve shape, right? It either flattens that curve down or it brings it up depending. So if you really want to know accurate demographic inference, unfortunately, sacrificing a couple individuals, especially for the smaller butterflies, I think you kind of got to get their thorax. But we did prioritize collecting old males, and for the
Hey, what are you talking about, old males?
yeah, we did.
One of
Hey,
your
I'm on
most
my way
good.
there, man. Um, yeah, in the genome assembly, we built it from a female, actually, just because Felix and I we messed up our samples and we meant to make a male because it's the heterogametic sex, but then we accidentally put all those ones in the wrong solvent, so we ended up using a female. So that was kind of funny, but uh, but yeah, I hope that answers your question in terms of a lot of the conservation. Like there will be people that tell you that, oh, I get whole genome data from just soaking a leg. It's apples to oranges. It's not the same kind of data. We're doing, like, and I'm not saying one's wrong. They're just targeted at very different analyses. But sorry, I do want to add one more note, actually. I'll just keep going. In the old days, like the old Euphidius days, some scientists were pretty renowned for doing genetics and going out and cleaning out a whole population because you needed a lot of individuals to basically characterize the genetic diversity of a population. And that's because imagine if you're doing something like what we call AFLPs or microsats. That's like trying to figure out how big somebody is by measuring the thickness of a fingernail. You get so like, whereas a whole genome, you're taking every measurement imaginable. You get so much more information out of single individuals that we could have done, like we did this whole project with 15 individuals. That would have never been possible before because we would have had to do 150, and that really threatens endangered populations. But we can do it with just a couple these days.
And was the Park Service cool with your sampling the number that you needed?
Yeah, it was their idea actually. So they were like I will say that working with them, that was the value. We didn't ever actually have to approach them and try to convince them of the utility. And this project really changed my perspective. Parks were there, Rob Sissons was there, the zoo was there, every single meeting that we talked about, the analysis, the project. So it's just like there was no convincing. It was all collaborative and
Oh,
equal
fantastic.
contribution. So yeah, it was good.
That's the way it should be.
Yeah, yeah. But you need good people in the parks, right? So
Yeah, it's been kind of crazy with the island marble sometimes. Not that there weren't good people, but there were too many agencies,
yeah,
plus Xerxes, the state, the feds, and individual egos that get into it. So it's been
yeah.
kind of a three-ring circus with the island marble for decades. But
Well,
it's getting there. But it sounds like you were collaborative from the get-go.
yeah, it was a lot easier that they approached us. So they already had it, and Felix's name kind of carried us through there, which was nice. Um, but in the US, I've noticed that for any point location, you have a lot of overlapping legislative boundaries. Like something can be a national park and it can also be a wilderness area and it can also be this. Yeah. And it's just difficult. Whereas in the Canadian system, it's just a national park and it's not even provincial lands. The province doesn't have any authority over it. So having one regulatory body to work with really streamlines it. It's difficult when you're doing more broad scale landscape genomics and you have to do multiple parks, but the fact that this was just all in one really it gave the park ownership, and I think they're more incentivized to really see this through.
Well, young Zachary, this is a strange country.
Yeah, yeah, but it's
We have
an
many
amazing
we
country.
have we have many jurisdictions and they're all defunded at the moment. So
Yeah, yeah, it's a lot of changes right now.
So I have a question about the ants. Is somebody doing some follow-on work there? I mean, that seems like an interesting question in and of itself that they use different ant species.
And so we were really lucky in that we were about a year and a half into the project, and Calgary Zoo started hiring a new coordinator for our project because they were helping fund it and stuff like this. And they hired an ant person that we knew, James Glacier, and he on his own for his master's doubled the number of ant species that were known in the province.
Oh, wow.
So
Wow.
I was thinking, like, just by accident, you couldn't have hired a better person because you just have a gestalt for it or you have an eye for it. And people were having a really hard time finding the larva for one and finding the ants associating with it. And he figured it out in about half an hour. So it was really lucky that we had him to ID the ants and to find the larvae. He just has a great eye for these really small things. And, the galleries are fantastic. Like you pull up a host plant is how you have to search for them. They're doing all kinds of larval surveys. Like the survey effort on the fan now is quite extensive. John Acorn's son, Benny Acorn, is doing his master's on this right now.
Oh, wow.
They're doing larvae surveys where they're pulling up all the lupines, kind of like you can kind of tip them over and then you can put them back. They don't really seem to be harmed at all. And they have ants underneath them. And a lot of the time you find the larvae there. And it's cool because there's the silvery lupine and the silky lupine that are both on the fan. And they only feed on the silvery lupine, even though the silky is there and the butterfly feeds on silky. The semiluna feeds on silky across the rest of its range. So it's a weird combination between ant and host plant and soil. So yeah.
Do the ants build any retreat for the larva, or do you know?
I don't know actually. We should get on the horn with James Glacier. I mean, they're as far as I've seen, the ants are just kind of in the galleries when you tip it over, and they're of course being tended to by the ants. And James has observed the larvae secreting honeydew, so it's not just pure pheromones.
Is James writing a paper right now, or is there a plan to publish some of this work? It sounds really interesting.
Yeah, we just put kind of like a natural history note in review somewhere that's led by Benny. So hopefully that'll come out soon. A lot of the information was actually in the species description, but it also has some detailing of their survey methods and stuff. It was kind of an awkward paper because it was like it was kind of all the information in the species description, but some additional stuff and just put in a more straightforward way. Here's the natural history. Here's what you need to know. Describing those survey methods without really reporting the results. So it's kind of a halfway piece between Benny's more substantial thesis work that'll be his like survey results. So they put transects all over the fan. And so we'll get to know, because really we need a habitat suitability model. We have one. I've built them the very coarse scale where you basically predict within grid cells that are 1 kilometer by 1 kilometer how likely is the butterfly to occur there. But within the fan, they really sub-select areas. And Benny's hilarious because he's like, well, this is old growth fan, he calls it. And it's where the lupines are just chunkier, and they really like that. They don't like the new growth fan. And so there's a lot of fine scale habitat selection going on. And then relating that to spotted knapweed's going to be important so that the parks, when they can have a truly an evidence-based approach when they're attempting to just eradicate this. But I have been to areas in Glacier searching for alternative fans where there is no knapweed control, and I was actually quite surprised that the knapweeds did just take over the whole fan. I was kind of like, whoa, okay.
So Zach, yeah, if Benny is hilarious, then he's really a chip off the old block.
Yeah, he's awesome. He's
I
great.
don't know if the members know John Acorn.
Yes,
Hope
we've
you
had a
do.
talk from him. It was really great.
Yes, yes. And he's done good books on butterflies of BC and Alberta. And he's a wonderful performer. Just a great man. So I'm so glad to hear his son is in this too. But I wanted to ask you, does the fan have a large population of Icaricia icarioides, the Waterval's blue?
Yeah.
And
No
does that
Icaricia.
complicate your survey?
Yeah, no Icaricia that I know of on the fan.
Oh, really?
Yeah.
Oh, that's really useful in terms of your survey.
Oh,
Because
yeah.
if they were there, that could complicate your survey.
Oh yeah. Yeah, and it's funny that you bring it up because I've accidentally collected so many carioides that I'm starting a project on it because I already have such good sampling for them. So, so I just got bags of these things because it took me a really long time. And Caitlin, I think I had a chat with her, and she kind of helped me, like, pick them apart a little bit. But
I
yeah,
still mix up an individual now and again.
oh yeah. That's yeah, it's amazing because they're really not closely related. So it's
Not at all.
yeah.
Well, Caitlin has determined some really good field marks. The way Jeff Glassberg used to find new field marks for certain butterflies in the East, and he really did. And Caitlin's got a wonderful eye that way for spotting discriminations that not many people had noticed before. We tried to get those into the field guide as much as possible, but you need those people with that good eye.
Yeah.
And,
Oh, speaking of
Caitlin
that,
teaches me how to tell those two things apart sometimes.
there was a really good one that James Glacier came up with that is actually bomb-proof. I should have brought this up. If you look at the chitin color on the thorax, brown in semiluna, black in Boisduvals.
Oh, cool.
And so that's just an ant guy thing, or an ant a person thing. I would never think to look at like you have to look beneath the SETI, but it's a really reliable character in addition to the blue flame.
Well, butterflies have wings.
Yeah,
They're
I know. They get
beautiful.
distracted. I get bamboozled by them.
So,
Talk about
as a non-expert, can you explain exactly where that is, Zach? I'm not.
Yeah. So basically, where the wings articulate, I just pinned up a swallowtail. Basically, right exactly where my pin went in there, in the center of the thorax, so just between the wings. Oftentimes, you'll see that there's some setae, which are the fine hairs, and they're worn off there. There's a little bald spot there a lot of the time, and it's shiny black in Boisduval's blue, and it's kind of like a dark brown in semiluna, as well as the curiously isolated hairstreak. So, but it is difficult to see because you got to use some forceps to gently spread the wings because they always want to keep their wings folded above their heads. So, in that
that's a
respect,
good one. That's a good one.
yeah.
Talk about cool names. James Glacier. Is his last name really Glacier?
No, I spelt it wrong on a paper one time. It's not. So it's a glacier with a S. Yeah,
That's a cool name anyway.
yeah, glacier. Yeah. Yeah, no, he's a good person. And if you ever want to go up and see the fan, he's up there. There's Rob Sissons, James Glacier, and Benny Acorn. They spent a lot of time on the fan there, and I'm sure they'd meet you all up there. And if you ever find your way up there,
Well, Carolyn, was it you who was suggesting a field trip?
do it.
I did, but I've been staying closer to home on field trips.
What about a
I
WABA
was
conference?
I was looking at the Seattle Pacific Science Center or the Woodland Park Zoo because they have.
Let's go do one at the
I have
fan.
trouble getting to Eastern Washington, let alone Waterton or
Let's go-
wherever.
Let's go do one at the fan, and then we can stage a mass defection.
Well, yeah, I might be interested in that if the
Sorry.
Canadians will take me.
Oh, they'd love you.
Well, I looked at those pictures and I thought, oh man, that may be my next road trip. Gosh almighty, it's just so gorgeous there.
Yeah, it's pretty good. And Parnassus smintheus, which is cool. It flies on the fan about 2 weeks before, and it's really surprisingly low for smintheus. I mean, y'all get Clodius at low elevations, and it's kind of more at the elevation that you would expect to find Clodius. But so it's just cool to find it in a low elevation spot like that.
What sedum are they using?
Lanceolatum. Yeah, I think. I mean, that yeah,
Well, that's likely. Yeah, that's
Yeah.
likely.
yeah. I'm not. Yeah, but
Have you ever smelled their pheromones there?
which
The male Smintheus.
no.
Check it out. They smell like Fritos. It's true.
Wait a minute. You're telling me you smelled a Smintheus?
A million times. I showed my butterfly faces slideshow the other day at Grange. This is cool. We had a movie night at Grange, and our Grange master, Chris Novoselic, came up with this 1960 film strip of Lincoln Browers about the courtship behavior of the queen butterfly, which was very cool stuff. With them putting out their hair pencils from their abdomen and stroking their anticonia and so on. Cool, very old movie. And then I showed my Butterfly Faces slideshow, which a lot of you have seen, 80 pictures of people with butterflies on their noses, and some music by Dire Straits. I hadn't showed it in 10 years because it slides, but I did it. And then I was remembering that 4 or 5 of the pictures are Smithias on people's nose in Colorado. And every single person remarks, that smells like Fritos. And they do. Male Smithias smell like Fritos.
Can I quote you?
What?
Never mind.
Yeah, you can quote me.
Okay, any other comments, questions?
I guess before we get off here, Zach, I think you still have my cell phone number, but for getting an address to ship you those labels.
Yeah.
My email's on my blog. That's the northwestbutterflies@gmail.com. That's probably the easiest one to remember, but
Okay. No, that'll be awesome. So they're setting me up with FedEx account this week. So
okay.
Which could mean next week, just based on how how things go. But no, that would be fantastic. So freezer's almost up and running. But yeah, I mean, if anybody, we got projects on, been collecting Parnassus Smithius group for the last kind of 4 years, and I've collected probably 300 of them. I can't believe I've never smelled one. John and I were just having a chat that, the McKayon group, Dodai specifically, I mean, they just smell fantastic. We talked about that for a while, but I never smell Parnassus, but
Male Eurymedon smell nice too.
That's right.
really, yeah.
Yeah, they smell like especially over on the east side, they smell like the Ceanothus.
Ooh.
Oh,
Yeah.
But the interesting part about the Bairdii group or the Dodai is that you get them right when they close and they still smell like it. So the Ceanothus, is that after they land on it, or do you think that it's something they're
No, I think it's just every one that I've ever caught smells like it. Some a little bit stronger than others, but yeah.
You mean like the Ceanothus velutinus?
Yeah. Yeah. It's like that. It's kind of hard to describe. It's not like peppermint. It's just like a strong plant smell, but yeah, it's
not altogether pleasant.
I like it.
like it,
It's strong.
but I'm a little weird.
Oh, really?
Well, not as weird as you.
Hey.
So Dave, you've unmuted. Did you have a question or comment?
Dave, that you? No. You're unmuted.
Yeah, I was just wondering, you unmuted and you're on screen, so I just wondered if you might have a question or comment.
You're talking
Yeah.
well, when you were talking about host plants, I went back and read our accounts of when we were raising semiluna. The one in the low elevation shrub steppes, very dry country, and they were feeding on Lupinus lepidus, which is a common plant in eastern Washington here. Not sure what David James found his on at the higher elevation. I know he raised some of them on Lupinus sericius, but I don't know that he found them on it. When the larvae pupated, the ones who were feeding on lepidus down in the low elevations, interestingly, they were in many cases they were finding an interesting place to pupate. That's on what is the name of that plant? I was just looking for it here. John, you know where we found them on
a whiskey dick?
Sorry?
Whiskey dick?
Well, it was in the Venus, but I'm talking about the plant that they were found under.
Astragalus, right?
Astragalus pershii.
Pershii, yeah.
And for some reason, they seem to like to go there to pupate, even though they're feeding on the lupin.
Well, it forms a mat close to the ground, so there's a shelter built in, you know.
That's right.
And do you think that they were ant-associated, like Camponotus or one of the bigger ants?
And the low elevation, they were ant-associated, yes, while the larvae were growing. I did see that on several occasions. I don't know if that was documented at the high elevation by David James. I was just running through this text to see if he had mentioned that, but I don't see it.
A great many places where semiluna is found in Washington. It shares food plants with Ichoroides and Glaucopsyche lignimus too. And they each have kind of like a different kind of modus operandi and a different sequencing as well. You know, the Satyrium diaposes eggs, and I think the others are half-grown larvae. So the Satyrium are always a little bit farther behind.
Yeah, yeah, yeah.
Well, thanks for, I saw you looking at that book. I knew you were digging up something, Dave. So, so
Yeah.
thanks for sharing with us.
I sort of remembered, but I didn't completely remember what we found. I knew that we're finding these differences between the low elevation and high elevation, and they were just puzzling the heck out of us. So that it was, there were fairly serious differences. We struggled with that quite a bit.
Sorry, I was just trying to see if I had your book on my shelf. I just got another copy of it and I was very proud to show it off, but I think it's at home. But what a work, like what a masterpiece. Holy hell.
Yeah.
That's this one.
Yeah, yeah.
I always tell people I know Dave Nunnally.
Yeah.
I don't mention you.
Yeah.
Touche.
Okay.
Zachary, first
Go ahead.
first rate work. Thank you so much.
Thank you so much. Yeah. Well, it's so nice to meet all y'all. You guys really carry your reputation all the way down in SoCal and all the way up in the Canadian North. So it's nice to finally put, the faces to names, and I really appreciate you taking the time to talk about hair streaks with me. That's
Oh
awesome.
man, that was great. Hey, love you, brother.
Cool. Yeah. Well, I'll see you in the field next year. I'm going to be coming up your way to catch blues. So I'll be sure to link up with y'all if we have a meeting at the same time. That would be great.
Where are you going?
I don't know yet, but going to meet up with Ross Winton, who I was hoping would be here, and I think y'all are on my way up. So probably the Olympic Peninsula.
What kind of blues? You're looking at Annie's blue, the Fuca blue?
Yeah, the Fuca Blue, just trying to figure out what some of the isolates are for Eukaryoides. I think we're going to do something like very much aligned with what we did here, but for all your funny blues up in that neck of the woods.
Oh, it'd be wonderful. I just saw Ann Potter just the other night. She came to a reading of mine in Olympia. I hadn't seen her for years.
No kidding.
And I said, what do you know about Annie's blue? How's it doing?
Yeah.
Otherwise known, John calls it the Straits blue or something. I don't know. Got different names, but it doesn't have a subspecies name because we haven't been able to discern any difference, even though it's got the weirdest, most distinctive ecology of practically any butterfly in the state. And
Really?
so if you can find something for us, go get them. Bravo. That'd be wonderful.
Yeah, if you guys have any notes, any information, or even if you guys want vials with, ethanol in it to go bag some, I'm always appreciative because it's just like the local knowledge really runs the show, and I'm just the lab jockey in all this. So it's I'm really interested in the hypotheses y'all have in terms of what the important isolates up there are. So I hope we can talk about that again sometime.
Are you going to start working with euphalotes at all?
I haven't got the Euphaloides bug yet, but mostly
No,
just
Merrill Peterson's doing that.
Yeah.
He doesn't have the kind of lab though.
He's
Yeah, he
working with
does.
people. Yeah.
Kind of lab that these guys got. That'd be great.
yeah.
So Allison, do you want to unmute?
What's that?
I can, go ahead.
Sure. I just want to say that Karen Holtrop, I don't know if she's on tonight, and I work on the Olympic National Forest. So
Okay.
if you're looking for a contact there, you can get in touch with either of us.
That would be awesome. That sounds like I'm not familiar with the geography, but based on the name, it sounds like it's right smack dab in the middle of where we'd be working. So that's great.
There's
Well,
Karen
We
right
did find
there.
Icarioides last year in a lower down elevation. So yeah. And I know Ross, so I can let him know.
Yeah.
Karen, when you say lower down, where?
Well, the little Quilcene trail near there, the 20 road.
And at what elevation?
Less than 3,000, like 2,000, 25, like 2,000 feet.
Great, that'll be another one like Taylor's checker then. Do these mid elevation things owe their affinities to the upper things or to the rare low elevation things?
Yeah.
Hey, Jessica. Well, in 1959, a federal judge ruled that the U.S. Post Office could no longer confiscate copies of this.
Okay, I don't know who's got their radio on, but if you could mute, that would be great. Thank you. Go ahead.
Well,
Sorry,
anyway.
Karen, just the interruption.
Zach, if you're doing Washington blues, it would really be a service if you could give this full genomic treatment to the Spangled Blue, the endemic in the High Olympics, because Dave's been doing he did older and he's done some recent dissections, and they share some affinities genitalically with Acmon and some with Lupini, which it is considered in the catalog. And man, wouldn't it be a good thing, John and Dave, if we could get a full genome done of the
Spangled blue.
Spangled Blue?
Oh, yeah.
Absolutely. Mm.
Yeah, and Carter Ernest would also enable that. He's in the national park.
Yeah. No, this is, well, if it's closely allied with the Acmon blue, we were thinking about maybe doing some work on that. Yeah, let me take a look at that. Yeah, it's a lupine eating thing, right?
Yeah.
Lupine spangulatus is the current taxon.
Yeah.
Oh, okay.
We don't
Yeah.
want to avalanche you either,
Well, that's what my department tells I'm supposed to be doing right now. I'm just supposed to be getting avalanche. So yeah, so y'all are doing me a service.
Well, all right then.
Yeah.
We'll send you more information about both the Straits Blue and the
Yeah, just a priority
uphill.
list would be really helpful. That'd be great.
Good.
Yeah.
Gotta get these things figured out.
Heck yeah.
Allison, did you have another comment?
No, other than to say the Olympic Park National Park is in the middle of the peninsula, and then the National Forest is around the edges. But it all started as the National Forest, and then the park got carved out.
Carved out.
Out of it. So that's sort of how the geography goes.
Okay. That's awesome.
Okay, well, we're looking forward to seeing you next year, we hope, Zach. And thank you so much. It was just a wonderful talk. You did such a great job of explaining a very complicated topic that a lot of us don't know much about. So thank you. Really, really excellent. Really appreciate it.
Well, thanks, Paulette, and thanks so much for reaching out and organizing all of this. That's just so kind of you. You did a fantastic job. So I'm really thankful for all of you for your time, and looking forward to seeing you in the field again next year. That'll be awesome.
Yeah,
Come up for my
serendipity.
free pizza.
There you go. Okay, so our next month's talk is by David James, who you also met, Zach. And he's going to be talking about the program that he has underway to put the little mini trackers on the Western monarch population and see if they can start figuring out where they go. So that'll be that'll be our May talk, and that'll be our last one for the season before field season starts. And we hope to see everybody out in the field. Okay, so thanks, Zach, again, and thanks everybody for being here, and we'll see you next month.
See ya, everybody. Bye-bye.
Okay, good night.
Thank you.
Take Take care.
Happy April Fools.
Yeah.